The genus Streptococcus is another group of medically important gram-positive cocci. Streptococci can be classified based on their genus and species name, their hemolytic activity, or on the presence of Lancefield antigens on the surface of their cell wall. Looking at the genus and species of the Streptococci of medical importance, we have Streptococcus pyogenes, Streptococcus agalactiae, Streptococcus pneumoniae, Streptococcus bovis, and Enterococcus faecalis. Enterococcus faecalis used to be known as Streptococcus faecalis, but because it lives under very different conditions than most of the Streptococci, it was reclassified as Enterococcus. As we look at classification based on hemolysis, we need to make sure that the organisms are growing on blood agar. Alpha hemolysis is an incomplete hemolysis of the blood that causes the agar under the colony to appear green. You want to make sure you're looking at the agar and not the colony color. Beta hemolysis is a complete digestion of the red cells. Here, the agar around the colonies appears clear. These hemolysis types are sometimes best seen by holding the plate up to the light so light can show through it. Gamma hemolysis is when there is no effect on the red blood cells, so that the agar immediately around the colony appears unchanged. The Lancefield antigens are antigens on the surface of the cell wall. They were first discovered by Rebecca Lancefield. She discovered antigens A through M, but there are only 3 that are medically important. Lancefield group A is found on Streptococcus pyogenes. Lancefield group B is found on Streptococcus agalactiae, and Lancefield group D is found on Streptococcus bovis and Enterococcus faecalis. The alpha hemolytic strep do not possess Lancefield antigens. They are generally classified as viridan streptococci. Viridans means green. The identification process always begins with the microscopic morphology. All Streptococci and Enterococcus are gram-positive cocci. You then want to look at the macroscopic morphology. Many of the Streptococci are fastidious and require blood agar to grow. They typically show as punctiform to circular colonies that are convex and entire. They may be gray or white in color depending upon the species. It is important to note the hemolysis. Note whether it is alpha, beta, or gamma. This is an important feature to identifying the organism. It's also an important feature to help you set up the appropriate biochemical tests. The biochemical tests appropriate for Streptococcus include catalase, Optochin. Optochin should only be done on alpha hemolytic organisms. Bacitracin. This should only be done on beta hemolytic organisms. The CAMP test should only be done on beta or gamma hemolytic organisms. The high salt is done on all organisms, and the bile esculin is done on all organisms. All Streptococcus and Enterococcus are catalase negative. Additional biochemical tests are going to require incubation. You select the appropriate test based on the hemolytic reaction of the organism on sheep blood agar. The Optochin test uses a disc that has a P on it, so sometimes we refer to this as the P disc. You do not need an entire sheep blood agar to do one test, so you can divide the sheep blood agar into quadrants. You will then streak one quadrant for confluent growth with your test organism and place the Optochin disc in the center of the lawn, gently tapping it down. You incubate the plate for 18 to 24 hours at 35 degrees centigrade with the plate inverted. The Bacitracin test uses a disc that has an A on it. Again, you can divide a sheep blood agar into quadrants, streak one quadrant for confluent growth with the test organism, and then place the Bacitracin disc in the center of the lawn, gently tamping it down. This plate also incubates for 18 to 24 hours at 35 degrees centigrade with the plate inverted. The CAMP test uses Staph aureus. Staph aureus will have been streaked down the center of a sheep blood agar plate. You will then streak the test organism in a single streak perpendicular to the staph. So the staph is down the center of the plate, and you're going to streak your organism up to, but not touching, the staph streak. You want to be sure your test organism does not touch the staph streak. It will come very close, but it should not cross it. This is incubated for 18 to 24 hours at 35 degrees centigrade with the plate inverted. The high salt is a broth, so you simply lightly inoculate the high salt broth with the test organism and then incubate for 18 to 24 hours at 35 degrees centigrade. The bile esculin test is a slant, so you inoculate the bile esculin slant with test organism and incubate for 18 to 24 hours at 35 degrees centigrade. After incubation, you want to look at the Optochin test. There is a chemical in the P disc called Optochin that will diffuse into the agar. If the organism is susceptible to Optochin, it will not grow around the disc. A zone of inhibition of 15 to 30 millimeters is considered a positive Optochin test. A zone of less than 15 millimeters, there's no zone around this P disc, is considered negative. But a zone of 15 to 30 millimeters is considered positive, and this one runs about 21 millimeters when you measure across the diameter. Notice that this test is reported as negative or positive. The novobiocin susceptibility test was reported as sensitive or resistant. The Bacitracin test is also a disc diffusion technique. Bacitracin, which is an antibiotic, diffuses from the disc into the agar. Some organisms are inhibited by bacitracin in the agar and will not grow. In this case, any zone of inhibition is considered positive. On this side, we have a negative test. The organism grows right up to the disc. Here we have a small zone of inhibition. You do not have to measure this one. Just seeing any inhibition is considered a positive test. Once again, this test is reported positive or negative rather than susceptible or resistant. Staph aureus, which was streaked down the center of a sheep blood agar plate for the CAMP test, produces a beta toxin which will diffuse into the agar. Some Streptococcus species secrete a protein that reacts with the beta toxin, producing beta hemolysis. Because the two will diffuse toward each other, you get an arrowhead of hemolytic activity at the end of the test streak. This is considered a positive test. So here you see hemolysis all along the streak, but not a distinct arrowhead. Here you didn't particularly have hemolysis along the streak, but notice the very distinct arrowhead of hemolysis. This is a positive CAMP test. Streptococcus species are fastidious and not salt tolerant. Organisms that will tolerate salt grow in high salt broth. Here you see no growth. That's a negative test. Any turbidity in the tube is considered positive. For the bile esculin test, some organisms break esculin down and you get a black end product. So if the agar does not have any change, that's a negative test, but if the agar turns black, that's a positive test. So here you see a negative and a positive. Just to recap overall, some tests are not done on every organism. For example, Optochin would not be done on the beta or gamma hemolytic organisms. Bacitracin would not be done on the alpha hemolytic organisms, neither would CAMP. So these organisms do not have results. You should not have tested them. All of the strep and enterococcus are catalase negative. Only strep pneumoniae is positive for Optochin. Only strep pyogenes is positive for bacitracin. Only strep agalactiae is positive for the CAMP test. Only Enterococcus will grow in high salt broth. However, both Enterococcus and Strep bovis are positive for bile esculin. These two are separated based on the fact that Enterococcus is positive for both high salt and bile esculin, while Streptococcus bovis is negative for high salt but positive for bile esculin.